Good afternoon. This is the IOE Rough Cut, and today we have Paul Cross with us. And Paul is a wildlife biologist from the USGS, Northern Rockies Research Center. And Paul has a BA degree from the University of Virginia in environmental science and a PhD from Berkeley in environmental science. So that's his background in terms of education. His research focuses on applied wildlife disease conservation and management issues around the greater Yellowstone ecosystem. And I assume that's what we'll hear about today. He's done a number of things. He tries to integrate empirical data and mathematical modeling in a lot of his research and the effects of host behavior on disease dynamics. And currently is working on brucellosis, chronic wasting disease, canine distemper, and sarcoptic mange. Mange? Oh, that's mange. Oh, I'll be darned. I look at that going, I don't know how to pronounce that. Of course I did. So thank you all for coming. And
thanks, Bruce. So thanks for having me. Few new faces, few old faces. Hopefully I'll be able to tell you something new today. But what I thought I'd do today is sort of go over this longer arc of this story about brucellosis that we've been working on in this ecosystem for the last almost decade and a half. This is work that's done in collaboration with a lot of different people. So that's why you're seeing all these different folks up on the slide, a lot of different state and federal agencies contributing to this work, as well as students, postdocs, and technicians, some of which are here in the room. So I'll try to call them out as I talk about their work and boil down like a thesis into a single graph. By my host. Oh, I'm unmuted by my host now. Okay. So typically, I tend to work on how host behavior affects disease dynamics and control in a variety of different ecosystems. But today we'll just focus on brucellosis, which is caused by this bacteria called Brucella abortus. So it is a pathogen that in this ecosystem infects bison, elk, and cattle, and is transmitted by abortion events, which is something that we're seeing here. An abortion event, which then individuals go and investigate. Then during that investigation, they get infected. It causes reproductive issues, which then creates abortion events, and then the cycle continues. Historically, and in most of the world, it's a human issue, a human infection, and that's the reason why we initially controlled it in cattle. Globally, it's one of the most common zoonotic infections of people. In the United States, though, it's very rare today, but historically it was relatively common, and that's associated with the 70-year control effort that's USDA's worked on to control brucellosis in cattle. But still very common in any society where you're drinking unpasteurized milk and living in a pastoral society with your sheep, goats, or cattle. Within the Greater Yellowstone ecosystem, the historic story around brucellosis was basically about these two different issues. One was bison that held it at relatively high prevalence and then exited the park in two different locations in the western portion of Yellowstone National Park, and then coming out the north as well. But then this other secondary issue of elk feed grounds around Wyoming, 23 different sites in Wyoming where the state and federal agencies feed elk during the wintertime. And in those locations also had higher prevalence of disease in elk. So the story probably begins around this time in the early 1900s. Bison are at low levels, down to about 18 individuals in Yellowstone National Park. And at that time, they were held largely in the Buffalo Ranch, which is out in Lamar Valley. And at that time, we also had domestic cattle in the same area, which were there for milk, providing milk to tourists and to staff in Yellowstone, and were observed cases of brucellosis at that time. And that's probably at least one of the times when brucellosis was passed from cattle to wildlife early on. So that's where we believe it's a European disease, and cattle came over as we brought our cattle over, then came over into wildlife and into bison, probably around this time. And so a lot of the historic discussion has been around bison and bison leaving the park. Bison were at very low numbers and then increased over time. This is just the total number of bison in Yellowstone National Park, and then the number of removals. And for the last 20 years or so, we've been in this dynamic of increasing bison populations followed by large reductions at the boundaries that are both for sort of that are for disease control purposes as well as population control purposes. And so a period of large depopulation followed by recovery, followed by depopulation, and so on and so forth is sort of the dynamic that we're in at the moment. My work has not focused a ton on bison, and that's predominantly because there's only a few cattle ranches that are actually at risk of getting anything from bison. So most of our effort's been focused on elk, and that's where I'll have most of the talk today about elk. So our elk work started around the feed grounds, which was historically thought to be the main focus of any issues. But we had not actually looked at much of the ecology. Prior to us starting to work on it, it was predominantly worked on by veterinarians working on vaccine development issues. So one of our first I'll start to tell you why the feed grounds initially exist and why do they continue to exist. They were probably started back at the turn of the century. The National Elk Refuge, I think, has been going now for over a century. And initially started as a way of mitigating loss of winter range, but also elk were feeding on cattle haystacks. And so the state wildlife agencies were obligated to mitigate that in some way, either by chasing them away or by creating another haystack in a different location. And so that's essentially the origin story of a number of the different feed grounds. So that was the initial issue, but disease was not one of the initial issues. This then creates some aggregation effects of concentrating them by feeding them, which has then resulted in some disease problems later on, which is creating another need of why we separate cattle from elk, is because we don't want disease to transfer between the two, which is why we feed them in some places to keep them separated, which continues to create the disease problem, which is why we continue to feed them. And so then we're in this cycle of how do we get out of feeding while also keeping them separated simultaneously. And that's some of the dynamics that we're trying to work on around the feed grounds. So one of the first issues that we tackled around the feed grounds was just how are feed grounds different from one another. There's 23 different feed grounds, and at a number of them we have disease testing information from a number of different years. And when we just look at which feed grounds are fed for longer than other feed grounds and what's the prevalence of disease on those feed grounds, it seems like there's some relationship between feed grounds that are fed for longer and whether they have more disease or not. So, and then on this graph, the size of the bubble relates to the number of tests that we have at that site. So sites with few samples, we're less confident in our actual estimate of what the disease prevalence is at that site. So feeding for longer seems to be associated with more disease. And the way we put this together actually is that we initially thought that it would maybe be related to the number of elk on the feed grounds would be correlated with the amount of disease at that site. But actually this site here, this one, is the National Elk Refuge, which is what you're seeing in the bottom photograph. So that one's about 10 times more elk than any other feed ground. It's about 6,000 individuals, whereas the average for the other sites are about 600. So it also has one of the lower disease prevalences of any of the different sites that we've looked at. So the way we're interpreting that at that time, we'll skip that, was that we think that basically it depends both upon the density at the site, but also the timing on when you're at the site. And the reason for that is that we think that transmission happens later in the spring. Again, transmission we think is about abortion events. We think abortion events might be from March to May. So if you have some sites that are feeding throughout that whole season, then you're getting transmission occurring while the feeding is ongoing. So this would be like Dell Creek is a site that doesn't have a ton of individuals, but feeds for a very long period of time from January through April. But the National Elk Refuge has a ton more individuals, but also has the shortest amount of feeding time of any of the sites that we look at. Being in the snow shadow of the Tetons, it seems to dry out and become snow-free early. So it feeds for less time. So if we have transmission that's happening at the edges of that, then it's sort of like this area under the curve of when are they aggregated relative to when are they transmitting. And if they're transmitting later, then it might be okay that there's a ton of individuals on the National Elk Refuge for this particular disease. But Dell Creek might be worse even though there's less individuals there. So some of the dynamics that we're trying to work on is how might we be shifting this towards earlier seasons so that we can get feeding to be outside of the area of time that we're actually, where there's transmission. And so that's one of the field experiments that they're currently doing at a number of different sites, is trying to stop the feeding earlier than what you might do just because of the snowpack of that year. For some people, this is strange that there's some sort of experimental treatment about feeding. The idea is that we already control the feeding, but actually the feeding is driven by the snowpack of the year. Sites that feed later tend to be fed later because they're in areas of higher snowpack. So what actually happens on most sites is that the snowpack dries out, we get new green up, and then the elk leave, and then feeding stops. So the feeding is driven by the ecology of the area rather than the managers deciding when they stop feeding and then elk leave. What we didn't know at the time that we constructed this graph was we just sort of like suspected that transmission was later, but we didn't actually have the information about when transmission happened. And so then over the next 8 years from the time that we did this one, we did a follow-up study of looking at, I think this was about 800 elk over 8 years, where they tested elk, and if it was positive for disease and pregnant, it was then monitored through time to see when abortions occurred or when live births occurred and where those happened. And out of that, we had about 150 individuals that were positive for disease and pregnant. And of those 150, we had about 30 different abortion events. So a lot of effort for a very low frequency event. Out of those 30 events, we can then look at when those events happen. And it is roughly according to what we thought ahead of time, that it was mostly March, April, and May. By June, we're having all the calves are coming out on June 1st, plus or minus a week or so. So obviously, it's going to tail off there, but abortion events tend to be in March, April, and May. So feeding that's happening in January and February, probably much less important than feeding that's happening in March, April, and May. And one of the issues is that the feeding sort of cuts both ways, at least on the short term. If we reduce feeding so that we're not aggregating during the transmission season, then they're potentially going off onto cattle property and creating more cattle risk. So we're reducing elk-to-elk transmission by reducing the feeding season, but creating more risk on the short term. If we feed them for longer, we reduce the spatial contact and the contact with cattle, but we're increasing the elk-to-elk transmission. So we get into this problem of short versus long-term benefits of reduced feeding, whereby if we reduce the feeding and it does result in lower transmission rates, we've got the short-term impacts of potentially more contact with cattle. And we need to go through those short-term costs to get to potentially longer term benefits of lower elk disease prevalence. And that tends to be one that's not a great political dynamic of short-term costs for long-term benefits. But currently, we're still doing the field trial to figure out, can we prove that there are elk benefits to reducing the feeding season on some sites where we don't have conflict with cattle. So one of the studies that we've done around the feed grounds, and this was sort of like rich territory around the feed grounds just because it's both that we have 23 different populations that are operating a little bit differently and in a management style, so we can do some manipulations, and some of the capture is also much easier. So one of the manipulations that we did was a study with this student, Tyler Creech, who was a master's student here at Montana State. And what we did was we basically did a field experiment on the feed grounds where we fed them differently and looked at contact rates with fetuses in the different treatments. So the treatments were basically low density and high density feeding trials. So high density is what we currently do on the feed grounds. We just dump hay off the back of a truck in a normal way. But low density is we make them work harder and go over larger areas and distribute the hay out farther. And then we just look at contact rates using these proximity collars that record when you're within 2 meters of another logger. So we put one logger under a fetus and we put another logger on the individual. And then we just alternate one day to the next, treatment, control, treatment, control. And so what we find is that when we do low density feeding, we can dramatically reduce the contact rate between, presumed transmission units. We'll call them fetuses. I don't know what, so we'll do that. But on a day where it's just normal feeding, about 1/3 of individuals would contact a fetus on a normal feeding day. So this sets up our second field experiment as just to encourage the Wyoming Game and Fish Department on the state-run feed grounds to try to distribute the feed out at larger areas to the extent possible, and then see if we can not only observe it in the contact rates, but then observe it in the disease data itself at a longer time span. And that's stuff that's still ongoing. But has sort of highlighted to us the benefits of doing experimentation in the field, because it always brings up things that you didn't initially think about. And so one of our experiments was, again, to shorten the feeding season, because we'd had that correlation between feeding season and disease prevalence. The thought being, if we don't feed them, then they won't be aggregated. And then we'll get the benefit. So we know what the mechanism would be, but there's other things going on. So we think snowpack drives feeding, and feeding is driving brucellosis in this case. But actually, so what we're going to do is we're going to reduce feeding and see if that affects brucellosis. But the other problem is that aggregation is caused not just by the feeding, but by the snowpack itself and winter conditions. And so this is what the experiment is sort of highlighting to us, is that even though we control feeding, we don't control snowpack. And so by manipulating feeding, we may not get the benefit that we thought we would get on brucellosis, because what we're finding is that even though we're manipulating feeding, elk are still staying around the feed grounds and still are partially concentrated because it hasn't become spring yet. And we're not able to create, that as our experimental management strategy. So we probably will not get that benefit of what I initially thought would be a reduction of disease by 2/3, going from 30% to 10% by reducing the feeding season by about a month, which was what the correlation showed us initially. That probably won't be as strong in the field experiment as what we thought from the correlation. So I'm having to backpedal on that one a little bit. I was pretty pumped about it in the beginning. During the time that we were doing all this work on the feed grounds themselves, something started to change in the unfed elk situation. Historically, the unfed elk were presumed to be at very low prevalence. Around the other areas of the Yellowstone system, it was about 1 to 3% disease prevalence. And if you went outside of the Yellowstone ecosystem, it was basically not detected. So that was the thought that, well, elk are not good hosts for this disease, except when you concentrate them at feed sites, and then they can do it. Otherwise, it's basically bison are a good host for this disease, and they're a problem area, and that's the feed grounds themselves. But around the mid-2000s, things started to change in the unfed elk situation. Looking at just a couple different sites, this is Gallatin, Madison, elk seroprevalence data, as well as Cody, Wyoming. And what you can see is that it started at very low levels. This is what people previously sort of understood. Like, yeah, we get a couple positives here and there, but it's generally very rare. But around the 2000s, it starts to increase in both sites and is now around 10 to 20% in these unfed elk populations, which is about the same as what we get at the elk feed grounds, somewhere between 10 and 40% on the elk feed grounds as well. So there's a big change in the elk seroprevalence over time. And if you just look at these maps of where those increases have occurred in 1990, this is sort of what people thought, is that it's around the feed grounds, the disease, about 10 to 30%. But if we look elsewhere, it's relatively low. But if we estimate what it is in 2014, and this is work done by Angela, she's here in the room, it's in many different areas now than wasn't there before. And at rates similar to the elk feed grounds themselves. So the questions then start arising about why is that? Why are things changing? And part of those increases, if you take the rate of increase between 1990 and 2014 for each of those different sites, and here we'll just do it for Wyoming, and we plot that against the density of elk at those different sites, the rate of increase is itself correlated with the elk density at those sites. So it's increasing faster in areas where we have more elk. And we've subsequently done all this work about, well, are there different ways of measuring elk density? And are there some density ways of calculating it that are more correlated with it than others? At just many different spatial scales. And in all of them, in all cases, they're correlated, and there's no sort of one metric that's the best metric to be working on. So one of the longer term dynamics is elk are actually doing relatively well outside of the Yellowstone ecosystem. If you take a broader view of elk numbers, they're generally on the increase or stable, except in the northern Yellowstone elk herd and the Gallatin Valley elk herd and the Madison Fire elk herd. Those three, which are sort of like central to wolf and grizzly bear habitats, those three are down or lower than they have been historically. But everywhere outside is increasing or stable. And across the West, elk are at levels that we've never seen before. They're at high points for, since they were depopulated to very low levels at the turn of the century. So, for example, the Madison Valley, it used to have about 600 elk in 1970, and as of 2010, it was about 6,000. So that's a tenfold increase in elk. And in that same valley, we can get groups of elk that are about 2,000 individuals, which obviously we can't get a group of 2,000 when we only have 600 in 1970. So the group sizes themselves are getting larger at the same time as the number of elk are increasing. So the mean group size is still relatively small for elk. If you go out in forested territory as a hunter, you'll know that you don't see large groups. You see small groups of 5 or less is the mean. But in open agricultural areas, you can get these very large groups of several hundred to several thousand individuals at a time. So one of the things that disease people get really excited about is like, how does transmission occur and how does contact occur and how is that a function of density? And that's one of the things that we've worked a ton on. And we have two different sort of like frameworks for thinking about it and sort of modeling contexts. And that is one that like contact increases with the number of individuals there, which is sort of an obvious one, which would be related to like a flu virus that I have and I'm contacting all of you and all of you could potentially get infected, as opposed to another disease that might be like sexually transmitted where contact does not maybe increase with the number of individuals there. Your sexual contacts might be relatively constant regardless of how many individuals you happen to be seeing. The ELK system, when we measure that in a couple different places, seems to be a little bit in between those two different poles. It does increase with population size though, and with group size. So this is basically the indicator that when we measure contact, it suggests that the largest groups are probably disproportionately important in disease transmission. More contacts are happening there. And this is sort of the same thing that we're seeing on the feed grounds as well when we do the feeding trials of high and low density feeding trials and contact rates. So large elk groups probably much more important to the disease dynamics than average groups, but usually as ecologists we tend to work on averages as opposed to the extremes. But in this case, it's probably the extremes that are actually driving what's going on. So this then led to a project that Angela did. She's she's here. Was a PhD student at this time and is now a postdoc. So, but she was basically trying to figure out where are these larger groups and what can we understand about them? And so she flew across the eastern part of the Wyoming section of the Greater Yellowstone ecosystem looking for groups of different size and what are the covariates that explain where big groups are. And so this is the example of these elk groups that are of a couple hundred to a couple thousand. They tend to occur in flatter properties of open territory that tends to be agricultural land. So we have this convergence of large elk groups exactly on the properties where cattle would be, as opposed to large elk groups in forested, high terrain, high slope sort of areas. So that is not what we want to be seeing, but that's what we observe. So this is what that group size distribution looks like. Most groups are small, but some are very large. And if you look at how it varies as a function of wolf abundance in open and closed territories, if it's in a closed territory, it seems like large groups don't really change. They stay small. But in open habitats, both we get larger groups in open habitats, and there seems to be this correlation where if we have more wolves there, we also have larger groups there, which could be a chicken and egg thing. Like obviously wolves are going to be in areas where they have lots of elk, so they'll be attracted to large groups. But also elk could be responding to wolf presence by diluting their risk and remaining in large groups. We don't know which one of those is going on. So this is just the correlation that seems to exist. So coincident with these changes that are going on in the disease in unfed elk, we're also getting changes in cattle infections over the same time period. So the historic time period was that we had basically removed it from all cattle across the United States, and it was almost eradicated from the United States and cattle at around the late 1990s into 2000. We had a few spare cases, and then it was zero for a number of years. And then recently, as elk have been getting more infected in a wider range, we get a few cattle herds that are getting infected per year across the GYE. And this is like not many, 1, 2, 3 herds infected. So very few herds are getting infected. But the reason why we have this intense interest in it is not just because of those 3, 4 herds that are getting infected per year, but because if we don't control them, it could quickly spread to the rest of the United States and then erode the 70-year control program that's been going for going to keep the cattle herd clean. So the red states are just those states that have received cattle from those infected herds at the time that we saw that they were infected. So we move cattle across the country rapidly and all over the place. So when we move them, we need to know that they are negative, so that we don't start the infection there and then have to come back to it a couple years later and realize, oh, like now they've got disease and now we have to clean it up again. Now we would have to do that again across the entire country. So understanding where cattle are at risk and then knowing whether they're negative and can be moved out of the greater Yellowstone ecosystem is like a nationwide, cattle health issue. And used to be a statewide issue within the GYE. The rules within the states used to be that if 2 cattle herds got it within the state, then the entire state's cattle industry was basically on notice and additional testing requirements were required anytime they left the state. It's changed since then. Now there's this designated surveillance area that basically rings the Yellowstone ecosystem, and any cattle that are moved outside of this red boundary are then have to be tested and test negative in order to make it to other parts of the country, again, to keep the rest of the country clean. So now our issues are like, how do we draw this boundary effectively from a management perspective? And that also comes from the elk wildlife information about where are elk infected and where might they infect cattle. And it's a difficult one to draw because we've got this cost benefits of, if we draw the boundaries really broadly, then we're subjecting ranchers inside that boundary to costs of the testing when they might be at very low risk. But if we draw too narrowly, then we potentially have escapes where cattle are infected outside the boundary and there's no testing that happens outside the boundary anymore. So then the disease could go unnoticed for several years before we realize that we need to clean it up again. And then we're going to 30 different states and hundreds of different operations in order to make sure that everybody's negative. So just looking at that seroprevalence again versus where those few livestock herd cases have come from, it's sort of a hodgepodge across the whole ecosystem, but generally does correlate with where elk tend to be infected. We've had a couple different cases in Paradise and Madison Valleys. And if you look at it broadly, the feed grounds have about the same number of cases as the non-feed ground related areas, despite having higher prevalence for longer periods of time. So this is what gets back to the harder political negotiations about, shutting down the feed grounds. Are they protective or not protective? Well, they have enhanced the disease in that local area. They have probably spurred the transmission onwards into other regions, but for the local ranchers there, they are probably somewhat protective. But at a larger, broader scale, they're probably a net negative, fueling the disease in other areas. One of the projects that took several years to come to fruition is this one, where we did the genetics of Brucella across all the isolates that we get around the ecosystem. And this was done by a postdoc with us, Pauline Kamath here, but with collaborations across all the different agencies. And what we did was we just grabbed all the isolates we could get from that we had stored anywhere from cattle, bison, and elk, and then we whole genome sequenced every one of them. And this is one way of looking at some of those isolates. What we have here is each colored bubble is an isolate from the Yellowstone system. If it's not in a colored bubble, it's coming from a cattle somewhere else in the country. So what you're seeing here is that we have a number of different lineages in the Yellowstone ecosystem. But for example, for these two, we have a number of different isolates that are more closely related, like a cow in Oklahoma might be more closely related to that strain than the same strain, than a different strain in the same location also on the feed grounds. So we've got different strains that are really very, very different, which is probably suggestive of multiple different introduction events deep in time. That we had an Oklahoma cow move over to the Yellowstone ecosystem, put in one strain 100 years ago. Then we had another Arizona cow come in and give a different strain also 100 years ago. And now we have these two very different strains in the same location. So we see at least 5 different introduction events within the Yellowstone ecosystem. I think since the time that we did this one, there's another lineage that's also different from these 5. And what we can do with this information is a number of different things. One, we can look at transmission rates between species in different locations, as well as how these lineages have spread through time within the Yellowstone ecosystem. So this is what the phylogeny looks like if we root it in time. But each different color is one of those 5 different lineages. So this lineage 2, this purple one, is this guy here, which is predominantly Yellowstone bison lineage, with a few elk in the northern Paradise Valley. So what this suggests to us is that the strains that we have in Dillon and western Montana are unrelated, completely unrelated, and separate introduction events than those that are in the bison themselves. So this relates to management operations where livestock agencies have wanted to push for vaccination of bison. And some of the logic behind that is that that may then have ripple effects outside the boundaries of the park where bison are infecting elk. And so if we control it in bison, then it'll go away in elk and we'll get the ripple benefit. But this suggests that that is not likely to occur, that any control in bison is probably only going to help it in bison and have no impact in outside elk populations. And those outside elk populations, they get these strains derived from the feed grounds themselves. So what we can do is we can take that phylogeny and then sort of like superimpose it in space and take different slices of time. And so what we see is that when we go back in time, these isolates are all observed to be originating from the feed grounds and then spread to outside. The bison, stays in bison. But through time, we can see them seed into new areas in western Montana. So what we think is that the feed grounds have sort of been the fire that have sent off sparks into these new areas where elk are, and they're at higher levels than they've been observed historically. In the 1990s, those sparks were probably flying off the feed grounds as well, but the group sizes and such were not sufficient for the diseases to take off at that time. But that in the 2000s, we're in a new regime where sparks are flying off and the disease seems to be taking in areas that hasn't been historically. And if we measure what that diffusion rate looks like from the genetic data, it looks at about 5 to 10 kilometers per year over the whole time span. But if we look at the most recent estimates in time, it looks to be about 12 kilometers per year, which seems relatively slow, but this thing is one that could creep up on us, where if we take that and project it for the next 20 years, say, we get an area, the DSA would then become an area about the size of California, covering most of Montana, Idaho, and Wyoming, getting down to Salt Lake City and into Colorado. So that's a major beef producing region of the United States that we have no real tools of how to combat this disease within the wildlife reservoir. We can also use this genetics to infer what the previous hosts were and look at whether cattle herds are themselves infecting one another or they're independent introduction events from elk or bison, which is important to know whether we're on top of the cattle situation. If we're on top of it, then each one's an independent event, as opposed to it's unobserved in cattle and one cattle herd's being moved to another one and then infecting it there. So the way it would look is if there's only within species transmission, like you'd get something that looks like the one above. But if there's lots of transmission going across species, you'd get a phylogeny where an elk isolate looks like a bison isolate, and then the same thing on a different part of the phylogeny over here as well. So there'd be a lot of mixing of different types of hosts within strains that look similar. And then for the cattle case, what you want to see is that cattle infections look to be very different and are in different parts of the tree, as opposed to cattle herds are related to one another more closely than they are to wildlife, which is suggestive that it could be that cattle herd 1 gave it to cattle herd number 2. So that gives you the setup of how to interpret this graph, which is then the actual information. So the greens are cattle cases, and all of these cattle cases, with the exception of one, is a cattle herd. In one case, it was 2 herds that are more closely related to one another than to anybody else that we have. So that could be because one herd gave it to another, or because there's wildlife isolates that are unsampled that are actually somewhere in between them. But it looks like for the most part, each cattle herd is infected from a new strain coming over from wildlife. And for every one of them, we think it's elk related and not bison related, which we sort of knew from the epidemiology of where the cattle herds are and where are the bison. But this is showing it to us again with the genetics as well. And we can also see that there's transmission going back and forth between bison and elk as well. And it's not surprising on the National Elk Refuge, where we feed them in the same location. So that is unsurprising, and we observed it there. But we also observe it up here. This is that Yellowstone bison, not the National Elk Refuge bison group. But even within this group, we get transmission that's happening between bison and elk. So it happens both in a fed situation as well as an unfed situation, which makes it difficult to think about sort of like eradication programs and bison, which are probably like logistically feasible if we felt like it was socially acceptable. We probably could eradicate in bison, but we have this unmanaged elk reservoir that we think would then just transmit it back to bison again, which we would not control. And so it's hard to sort of argue that we should control it in one while not controlling it in the other. And we don't really have the tools to control it in either. Well, in elk. In bison, we can basically, if we treated them as cattle and did a test and remove program, it would be effective. In elk, the logistics of doing that are just beyond what anybody's willing to consider. This is what if we take that projection of 12 kilometers per year and project it out for 20 years, this is what we're looking at. Hardware Ranch is an interesting one to us because it's right outside the DSA in Utah. Would be the next state to get it. And it's a fed elk situation, which is right on the boundary of the DSA at the moment. So some of the things that we've been moving to now has been trying to look at cattle risk and how that varies in space and time. So what we know is we know a lot about how elk are moving during the springtime. We have a tremendous amount of GPS information from elk all across the Yellowstone ecosystem. So we know where elk are, and then we know something about the timing of transmission from the abortion event data. So what we're trying to do is put those two things together and then provide that to managers about where do we think elk risk is the greatest. We've done that in 2 different situations, one around the feed grounds and one around the Montana portion of the DSA. The interesting part to me here as a scientist is that there's all this interest in wildlife, in the wildlife field, that they work on where individuals are. Their main emphasis is describing how animals move around and where they're located in space, whereas the disease dynamics people are all about temporal changes over time. And so what we're trying to do in these studies is to try to merge these 2 different fields together of both the space portion and the time portion. But it basically comes down to a multiplication, which is not that hard to conceptualize, but harder to implement. But it's basically, we're trying to calculate number of elk on a per pixel on a given day, multiplied by the seroprevalence, and we know what the pregnancy rate is, and we know the probability of aborting given you're pregnant and given you're seropositive for the disease. So then that gives us basically the number of abortions that are falling down on a pixel per day in different areas. So this is what one of those simulations would look like around the feed grounds. All the elk are really concentrated here in the beginning of the year. These are all the 23 feed grounds. They don't go very far from the feed grounds in the beginning, but then once feeding ends, then they start to shift away from feed grounds in April into May and generally go upslope. So we can take this and then match it to the timing on when abortion's occurring to look at where are cattle most at risk in this ecosystem at what point in time. It turns out that, at least for the Montana section, I believe also for the Wyoming section, most of the abortions, well, I'll speak to the Montana section, most of the abortions are happening on federal properties, but we have very few allotments that are operational, so there's no cattle there. So most of the risk is actually happening on private property in the winter, as opposed to on the federal property, because on the federal properties, there's not any cattle there until much later in the season. This is what that looks like. If you look at the Montana section, this is the federal grazing allotments for BLM and U.S. Forest Service and when they turn on during the season. So we get a number of different BLM allotments that are outside this DSA boundary. But within the DSA, we have very few allotments where cattle are actually there until much, much later in the season. Whoops, sorry. And it's only like in mid-June when we start to get federal grazing allotments with cattle on them. So for this reason, one of the reasons why we think most of the cattle risk is happening on private properties earlier in the season. Thinking again about that transmission graph of when transmission's happening, it's mostly done by June. And then this leads us to our last study that I'll talk about today, which is one that's currently ongoing. One of the questions we'd had from this is like, how can it be that we have seroprevalence in elk outside the feed grounds where they're not being fed at similar rates as what's going on in the feed grounds? Granted, there are some large elk populations, but yet we're still concentrating them at really high levels on the feed grounds themselves. And so one of the thoughts was that when we'd done these trials where we put out fetuses and looked at contact rates, we observed that fetuses don't last very long on the feed grounds. We put them out there and they're gone in 24 hours. And that had been observed in other studies, but one study was done around the Yellowstone ecosystem, well, around Gardner and West Yellowstone, and they observed that fetuses were out there for 20 to 30 days. And we knew that the bacteria can be infectious for 20 to 30 days, depending upon whether it's protected from UV light and other things. So that was one of the thoughts, that was that, well, yeah, we are aggregating elk on the feed grounds, but because scavengers concentrate on the feed grounds, maybe they're removing the infectious material quickly, and that's what basically equates the feed grounds to the non-feed grounds as the scavenging rate differs. But we only had sort of like two different points of comparison. There's one point in West Yellowstone and Gardner, and then the feed grounds themselves. So then the study that we're currently doing is looking at that scavenging rate more broadly across the ecosystem. This is something that Kim's doing. She's here trying to check up on whether I'm portraying it correctly, and Victor's out in the field also working on this project. And we're in year 2 of this project. Initially, we thought it would all be about coyote removal. And we thought, well, that might also have management implications because coyotes are unregulated vermin that can be taken at any point in time, but might be a disease sanitizing agent. But also the federal government removes coyotes in some areas, as you know, when livestock ranchers want them removed. From Wildlife Services does that. So we thought that that would be an interesting angle as well. When we followed up on this, actually they don't remove that many coyotes around the counties of the GYE. It's in many other counties, they remove a few, but not that many. But also what we're finding is that coyotes are not the main one taking fetuses from our sites so far. That's mostly golden eagles and bald eagles. They take a third. seem to be teaching their young about how to take some of them, as well as a random assortment of other things. Bears seem to be like too slow to get there in time. But one of the strange parts that we're getting from this one that we didn't expect maybe is that the fetuses are removed in all places relatively quickly. It's like 24 to 48 hours for the most part. And so now we're sort of confused again as to why elk have it at such high prevalence in some areas. And it could be just those large groups, and all transmission happens within the first couple hours. It drops out in a very large group, and then transmission happens, and it doesn't matter whether it's there for another 20 days or not. They don't unlikely to walk over it again, and everything happens really quickly. That could be part of it. We're still trying to figure it out. Since we're the Bobcats, I'll leave you with the Bobcat photo and take any questions you have. Thanks. Kathy. We know that they can be test positive, particularly wolves and coyotes, I think, are seropositive, but we know nothing about whether they transmit, but I tend to say they probably don't, but there's no information on it. Nobody studied it. I think it's unlikely that they would, even if they did have abortion events, like that those would then be transmitting to cattle. It just seems like, I think that's where people would go with that comment, is like, oh, this is another reason to hate wolves. But I think it's really unlikely that they're a strong agent of transmitting to anybody. But they can get it.
Is there a way to test for to diagnose brucellosis without blood tests, or can you not observe it in a herd without extensive testing? Remove the diseased animals without.
Yeah, so we've gone down one tangent on this one where we've tried to do this with disease detection dogs, which is one of our more risky projects, which I think would actually work if we could set it up right. So the idea is that you could get fecal samples and then train a dog on what's a positive sample, what's a negative sample, and then do it like without actually catching the animals themselves. You would just run dogs in different places. And maybe that would be a way of detecting it on the boundaries where it's rare. It turns out that that's really hard to do, to set up the trial effectively, because we don't have controlled infection challenge trials with this one. And that's because brucellosis it was one of the first weaponized bioterrorist agents, I think back in World War I. And as a result of that, and 9/11, it was then listed as a select agent of like under high control. And as a result, there's only one place in the country where you can do a challenge trial on large animal with brucella. So what we would want for that disease detection project would be to challenge them with the disease, then we know they're infectious, and then we get the appropriate samples, and we can set up the negatives and the positives in the right way to train the dogs appropriately. And dogs have been shown to be able to detect cancer and all sorts of other things. So this is just one more step along the line. But what we're struggling with is that we have individuals that are positive on blood tests, but they may be positive because they were infected years and years ago and are basically recovered. And then we're training the dog on that one unknowingly and comparing it to a control. And so we're not able to really train the dogs on the right samples. So that's part of the tricky part. So long-winded answer to your question, no, we don't have a non-blood test related. Even if we were to get that one, like it wouldn't be sort of validated by the ag industry. It would just be sort of like for research purposes, probably. It would sort of set up the conditions where, like, if you observed it in the field in a fecal sample, you'd then need to go back and really test that area to confirm it with a blood sample, which is what people would believe in.
I have to ask a climate change question. So is there any evidence that the brucella will last in the environment longer in a more mild spring than in
So heat also tends to deactivate it. The climate-related work so that might be part of it, but I sort of think the climate stuff that's like probably more important is how snowpack is modifying where individuals are. And initially, when we did the initial studies about the variation amongst the feed grounds, like I think I titled that paper something like Climate and Management, because we had this very strong climate signal driving the feeding program. And then we're showing that the feeding affects the disease. So at that point, I sort of like jokingly said like, well, you know, climate change will maybe be beneficial for brucellosis in the case that it'll reduce feeding and elk will be distributed more broadly, but hopefully we'll get on top of it faster than climate change. I'm now having to backtrack on that one, that I feel like climate change is actually just as fast as what our management, you know, changes in our management structure around the feed grounds. So they're both, you know, probably having equal effects on shortening the feeding season. So I think it may reduce transmission a little bit, but I think it's still an issue that we're wrestling with.
Hi, Paul, can you hear Norm?
I can.
Thanks. I just wanted to observe that the veterinary doctor who was very active in the wolf restoration, Mark Johnson, suggested that the role of wolves in brucellosis would be to greatly lessen the amount of stuff spread around, because the digestive system of wolves would reduce greatly the amount of brucella that was in the sample they were eating on. And I also observed that, of course, wolves don't abort pups.
Yeah, so this is also related to that scavenger study of like scavengers sort of being sanitizers of the environment, which is certainly possible and something that we're interested in pursuing. You know, the other thing that comes up when people talk about like wolves' effects on this particular system, there's other literature that suggests like predation might be helpful for reducing disease in all sorts of different species. And one of the mechanisms for that is that as individuals get infected, they might show some morbidity, which then predisposes them to being consumed by predators, thus reducing their transmission time period. That's probably not the case with brucella. Like, we don't think that brucella causes a ton of morbidity in elk or bison, and probably doesn't do a ton to enhance their susceptibility to predation. But there's these other mechanisms that might be at play that we don't know how they're sort of coming together. And that is how wolves may be affecting elk aggregations. And then we have all this information about how elk aggregations are themselves related to this disease. So it's not hard to see some connection between like wolves affecting elk behavior, which is then affecting the disease itself. But that could be operating in a couple different ways. You know, my mental model for it at the moment is that wolves are, you know, basically helping to disaggregate elk in areas of high timber and forested areas, and that's partly let's not be detected by wolves and be in small groups. But as we saw from some of Angela's stuff, like wolves are correlated with very large groups in open habitats, which could be, again, this like, well, let's, you know, we're not going to avoid detection, so we might as well be in a very large group and, you know, reduce your probability of getting consumed by being in a large group. So it could be that wolves have different effects in different territory and different habitats. And there could be, again, like short-term behavioral effects that differ from the long-term demographic effects, where wolves probably reduce elk population sizes on the long term, which probably reduces some of the, you know, disease risk on the long term, but in the short term might actually aggregate them in areas.
Great. Thanks for that, Paul, and appreciate your talk, and particularly just illuminating us that nothing is simple.
Okay, we're gonna
Thank you.